time to have the talk about rhino stool and reproductive success. So first, I would like to briefly talk about the health of captive rhinos compared to wild rhinos. So wild rhinos are browsers. So they will feed on leaves from bushes, trees, they'll eat twigs, and they'll eat any fruit that's hanging from a tree. And they are also able to diet switch if they need to start eating vegetation that is available, even though it might not be their primary food source. Now, captive rhinos have a more controlled diet that is provided for them, typically by human handlers, especially in a zoo type setting. And so captive rhinos can end up having a microbiome that is similar to domesticated livestock. And this could possibly be from the feed they receive, if it's similar to feed given to cows or horses. And it could also just be due to the fact that they are coming into contact with humans. Captive rhinos can also have a greater abundance of glycolysis and amino acid synthesis pathways. This represents an animal that is not getting an optimal diet and is therefore lacking in macro and micronutrients. And captive rhinos have also been known to have diseases not typically seen in wild rhinos, such as hemolytic anemia, iron overload disease, and even obesity. So either way, the bacterial genre found in the gut microbiome of wild rhinos is actually being replaced in the captive rhino. And obviously having a healthy gut microbiome is important to animal health, especially for those that are endangered or critically endangered. A healthy animal could potentially live longer and hopefully be reproductively successful. And so one aspect of this healthy gut microbiome / reproductive success that I will briefly discuss is the idea that certain bacteria are associated with phases of the female reproductive cycle of the eastern black rhino. So what is the relationship between reproductive success and the gut microbiome? So this slide shows some of the results from the paper that I looked at. The authors looked at bacterial genre, progestogen, which is a hormone that binds to progesterone receptors, and glucocorticoid metabolite concentrations. From stool samples from 16 female captive black rhinos with an age range of 5 to 40 years over a 21-month period. So out of a possible 253 genre, the study found 7 with significantly different abundance in the gut microbiome according to breeding success. 5 of the 7 were more abundant in breeding rhino, which you can see that in table 1. All of these genera, except for Certhia in the non-breeding female, had a lower relative abundance than the average relative abundance across all the samples. So this indicated to them that it was rare microbial genre. They were also able to identify 22 genera that were associated with the ovarian cycle, which you can see in table 2. 1 was associated with the luteal phase, 2 associated with the luteal and follicular phase, 5 associated with pregnancy, 7 associated with post-parturition or birth, 3 with pregnancy and post-parturition, and there was also additional 4 genera that were associated with the luteal phase plus a combination of 1 or other 2 phases. And they also noticed 4 of the 5 genera associated with breeding individuals were also associated with pregnancy, post-parturition, or both. And these were the Ericococciae, the Cronobacteriaceae, Solobacterium, and the Aetaphostomes. All 4 of these had a positive relationship with fecal progestogen, and 2 of these, the Ericococciae and the Solobacterium, also had a positive relationship with glucocorticoid metabolite concentrations. and all the genera associated with one or more phases of the ovarian cycle also had a lower than average relative abundance. So what would this mean for rhino conservation? So one of the ways this study could be used in conservation programs is to potentially develop these bacteria into a probiotic to help improve breeding success in captive rhino breeding programs. This could also be done through diet manipulation or prebiotics, but more work would need to be done to try and understand the feasibility in doing this along with the efficacy of this. Other things would need to be looked at, which would be any interactions between diet, gut microbiome, and the animal's physiology. So I hope this was somewhat informative for all of you.